Hi, so vlog number 3, and I want to talk about my first week doing my bachelor thesis. So as I mentioned before in earlier videos, I'm starting my bachelor thesis. I started my bachelor thesis now. I started on Tuesday, so today's Sunday. So it was right the weekend after my parents left, and yeah. Um, So I'm working in the neurogenetics department of my university, um, and I'm looking into Drosophila, which is a fruit fly basically, and I'm looking for specific neurons in their brain. Yes, insects do have brains. Um, they're pretty fucking tiny and a bitch to remove, but they do have brains. Um, and I right now this was my first week, so I started like learning all the techniques that I needed, and where everything is and how the lab works and everything. So I started breeding some specific lines that I needed, and one how can I explain this properly? So there are I don't know if you people know this, but there are specific, so you know that there are like luminous fish and everything, and they glow because they have specific protein that they can produce, which absorb light and then emit it again later. Um, and there in the I think it was 80s or 90s, I don't remember. There was a Japanese research team working together with the guy that found these proteins, and they managed to put the gene that encoded the protein and put it in other living species. And the interesting thing was that the other species also produced the fluorescent protein, which was groundbreaking because they didn't think that that was possible, and that there was like a ton of other things needed to produce the protein. So, um, so they did that and it worked, and nowadays like these fluorescent proteins are used for so many things, and to visualize things, and like even just in cells or visualize tissue and everything. It's crazy. It's used everywhere. So what I'm working with, it's called Flybo. It's derived from Brainbow, which was a genetic mice line which had a gene that encoded for, I think, 5 or 6 fluorescent proteins, all in a different color, obviously. And what you could do was you could switch the proteins around, so that how do I explain this? So you have this genetic line where the proteins are, and the first protein is the one that's going to be constructed. So if you have a genetic line and you start with a yellow fluorescent protein, then the cell in which this gene is encoded with a yellow fluorescent protein first is producing the yellow fluorescent protein. But you can flip the order around so that you have the red fluorescent or the green fluorescent, or I don't think there's blue, but there's like magenta. So you can switch it around, and you have like different fluorescent proteins in the beginning of the gene sequence. And depending on that, each cell can look differently, because you can't really control how you switch it around. You can just say, okay, I want to switch it, and then it just gets switched around randomly. So the hope is that for the neurons that I'm looking at, they each have a different color than the neurons surrounding them, which would make it really easy to, distinguish them, because with confocal microscopy you can just put on one laser and then shoot the, I guess, protein with just one wavelength, so that every other protein, every other cell is not showing. I hope that made sense. So yeah, that's what I'm doing. And for that, so I have to. The thing is the same thing for the flies, but it's just called flybow and has like different it's never mind, it's the same thing. So what I have to do is I have to create a fly that has the gene sequence with the fluorescent protein, that has a promoter. A promoter is like the starting point of the gene sequence, if you say, and the promoter, and oh yeah, the thing that switches the gene sequences around. So I have to create that, and in order to make that, I have to like get different lines and cross and breed them and crossbreed them, and then I have to like cross them again and then rebreed them, and it takes a long time. It's a process, and that's what I'm doing currently. So my actual bachelor thesis is not going to be what I'm doing right now, but it's going to be the same method, just with different lines. So yeah, it's a lot of fun actually, but I still have to like it, because I have to get the brains of the flies out, and I don't know if you ever saw a fruit fly, but they're like this tiny, like so tiny, and the head is just even tinier, and the brain is even tinier than the head, and it's just, oh my god. So it takes me about, like, I'd say 10 to 15 minutes to get one brain, and I'm going to be needing like a lot of brains to show that the thing is the neurons are where they're supposed to be, and that it's not just in one fly but like in multiple flies. And so I need multiple flies where it worked, and where the neurons are like different colors. And yeah, so I'm going to be needing speaking of flies, hello sir, okay, bye. So I'm going to be needing a lot of flies to prepare and to see if it worked. So yeah, that's going to be, yeah. So on my first day, on Tuesday, I arrived at 9, I think it was like 9:17, which was 2 minutes too late, because on Tuesdays and Thursdays we have seminars. So on Tuesday morning we have like, I'd call it journal club. So we are so the people are presenting like different journals, and like not just journals but like articles from scientific journals, and that's on Tuesdays starting at 9:15. And on Thursdays there's lab meeting, so we're like discussing the research projects of the doctorate and the master students within the lab. Yeah, so on Tuesday I was too late because I didn't know when to show up, because he didn't tell me. My supervisor is a doctorate, he didn't tell me when to show up. So I was like, yeah, I'm just going to be there at like 9:15, but then I had to walk because I left my wallet in my mom's car because she was visiting the weekend before, and then she drove home, and then she called me when she arrived home. She was like, yeah, you left your wallet in the car and it's here now. I was like, crap. She's like, yeah, I'll send it to you on Monday. And it arrived on Tuesday afternoon, of course. So yeah, that happened. So I had to walk and couldn't take the bus because my bus ID was also in the wallet. So I had to walk, so it was like 2 minutes too late. But yeah, so the whole the whole department of the neurogenetics, everybody was already in the room, and my lazy ass, I wasn't actually that lazy, but like my late ass walked in there, and so everybody was just like, oh, there's somebody who's late. And fortunate enough for me, our the professor of the neurogenetics department wasn't there, because she really doesn't like when people come in too late. Like, even she doesn't like it when people come in after the person started talking. She's like, she doesn't like that at all. She's really nice, but she just doesn't like that. And because I don't want to like make a bad impression or anything, I was a little embarrassed. But yeah, so I sat down there, and my supervisor was actually presenting a paper on different dietary routines that you have to go through, and like, because you need to feed the flies obviously so that they live and keep reproducing. Yeah, so that happened. And I'm going to yeah, that happened. I don't know why. I'm just rambling again. So yeah, that's a thing. So it's a long weekend today, Sunday. But tomorrow we have off because tomorrow's a holiday in Germany. Thank God. So I'm gonna be going back on Tuesday, and next week, next week's my birthday. My birthday is on May 25th, and I'm turning 22. And next Thursday, I'll be going home. Probably I might even go home on Wednesday, but that depends on how much work I have to do because I can't tell right now. Yeah, but that's gonna happen. So yeah, already 10 minutes again. This is where I sign off. I don't know if anybody's watching, but you know, have a great day and don't do drugs, or at least not harmful drugs. Yeah, bye.